Hi everyone and a very warm welcome to the first episode of the FARRE of this season, and we are going to begin with General Microbiology in Microbiology FARRE series. I welcome you all to this series. If you have any doubts, please keep writing to me and I shall make sure to answer them as well. So in the FARRE series, you know what it is intended for. It is the frequently asked, regularly repeated exam questions. That is, it is meant for revision, for a quick revision of the big chunks of the syllabus, so that you can get that confidence in exam and you can approach the exams with that much more of confidence. So today in FARRE, in general microbiology, what we are going to read is bacterial morphology, physiology, antimicrobial susceptibility, bacterial genetics, as well as AMR. Now, while these are the major topics, we are going to discuss a whole lot many topics today. So let's not waste any time absolutely at all. Let's quickly get into it, because in bacterial morphology, we will be discussing the cell wall as well as the bacterial appendages. In physiology, we will be discussing the bacterial growth curve as well as anaerobic methods, because that is a very important question which is asked, right? Then AST testing, all the methods, bacterial genetics, which is including antimicrobial resistance, the mechanisms of antimicrobial resistance, and of course we're going to discuss transformation, transduction, and conjugation, right? So let's quickly begin talking about bacterial morphology, in which bacterial cell wall is one of the most important asked questions. So you should definitely know the morphology. So if we start talking about, there are two types of bacteria in this world. There are the gram positive guys and the gram negative guys. So when I'm talking about a gram positive cell wall, I of course have to start with the cell membrane, right? So you know that what is this? This is a phospholipid bilayer, right? There is a phosphate moiety, lipid moiety, and the same is here, phosphate moiety, lipid moiety. So it's a phospholipid two layers. So phospholipid bilayer. What is it? This is a phospholipid bilayer. So you will draw this phospholipid bilayer and you will label that this right here is my cell membrane. Yes, this right here is my cell membrane. Now, everything which is above the cell membrane is going to constitute my cell wall. Yes, इसके ऊपर जो कुछ भी है वो मेरा cell wall बनेगा. It's very simple actually, because the cell wall of gram-positive bacteria does not contain much. Yes, so it only contains one major thing, which is the very thick peptidoglycan layer, and that is the reason you know that it is a why are the gram positive bacteria gram positive? Because they have this very thick and complex peptidoglycan cell wall layer, जिसमें कि आपका primary stain, which is the crystal violet, that gets entrapped along with the iodine and it cannot come out. Therefore, even after discoloration, you have the blue color retaining inside the cell wall, right? That is what happens. So that is why you know that the cell wall is majorly comprised of nothing but a very complex and a very thick peptidoglycan layer. Yes, so what is it? This is the very thick peptidoglycan. Peptidoglycan layer, and उसके ऊपर कुछ on top of it there are some things which help the bacteria in attachment to the host surfaces. So if the bacteria has to attach to the throat, to any part of the body, what is it going to use? It is going to use certain proteins which are called as lipoteichoic acid and teichoic acid. So what do these lipoteichoic and teichoic acid do? A, they help to connect the cell wall with the cell membrane, and B, they help the bacteria in adhesion to the surfaces inside the human host. So ये क्या है तुम्हारे? These are the lipoteichoic acid and teichoic acid. All right, teichoic acid. What do they help in? They help in adhesion and attachment of the gram-positive bacteria, and that's about it. That's it. This is the cell wall of a gram-positive bacteria. It is as simple as it gets. Now, what gets a little complicated is the gram-negative cell wall. Yes, what? Yeah, okay, so sorry. So what gets a little complicated is the gram-negative cell wall. So here we have a gram-negative cell wall. Of course, the beginning point is going to be a cell membrane. So what is it here? This here is the cell membrane, which is same phospholipid bilayer. Everything which is going to be over and above this cell membrane is going to constitute my cell wall. Yes. Now, the cell wall of gram-negative bacteria is comparatively a little bit complicated. Now you see, this is a cell membrane, a phospholipid bilayer. If you look closely, doesn't it look like I have copied and pasted this exact layer over here? Yes, if you ignore these red things for a minute, you know that I have exactly taken and copied and pasted the exact same cell membrane on top as well. Yes, so if I go from inside to outside in the cell, I will first come, so if there's a DNA etc., then the cell membrane and then the cell wall. So can I call the cell membrane as inner membrane and the cell wall as outer membrane? Yes, I can very much do that. So what can I do? I am going to call the cell membrane as the inner membrane. Therefore, this thing over here can be easily called as an outer membrane, right? So you understand that a gram-positive cell wall has no outer membrane. It only has a peptidoglycan, whereas a gram-negative cell wall has an outer membrane, which can be called as OM. Now, where did the peptidoglycan go? You know that gram-negative bacteria have a very thin peptidoglycan. Yes. So, let's draw that very thin peptidoglycan. So, why does it have a thin peptidoglycan? You have to accommodate so many things on top of inside the cell membrane. So, if on top of this an outer membrane has to come, this has to compress itself. Okay, so when it compresses itself, what happens? Let's see. Okay, over here, this is a cell peptidoglycan of the gram negative, very thin, extremely thin. It is almost 100 times as thin as the gram positive cell wall. So don't you think that when it has compressed, there is a lot of empty space which is there in the top and bottom of the peptidoglycan? There's a lot of empty space. So I have rightly labeled that what is it? This is my extremely thin, very thin peptidoglycan, and around the peptidoglycan there is lots and lots of periplasmic space which is present. What is this? This is periplasmic space. So if I ask you that is there a lot of periplasmic space in gram positive bacteria? No, there is hardly any space. There is no space over here. So mostly periplasmic space is almost nil to absent in gram positive bacteria, but there is a lots and lots of periplasmic space in the gram negative bacteria. और इस periplasmic space में क्या रहता है? There is a certain enzymes such as the beta lactams etc. which are present in this periplasmic space. Right, now let's look at this outer membrane. You see these red proteins which are there. What are these? These are proteins attached to the outer membrane. So can I call them as outer membrane proteins? So an outer membrane proteins can also be written as OMP. And you know what do these outer membrane proteins do? They come together and they make a channel. Is channel se kya hoga? Ions can go out and come inside. So these are like little pores in the cell wall. So can I also call them porins or porin channels? So porin channels or porins are nothing but they are the outer membrane proteins. So in which bacteria are porins going to be present out of gram positive and negative? Gram negative. So porins are mostly not there in gram positive bacteria because there is no outer membrane, whereas in gram negative bacteria, because of the presence of outer membrane, there is this outer membrane proteins which come together and they make a channel which is called as the porin protein. Yes. Now it does not end over here also. The gram negative cell wall is a lot more complicated as compared to gram positive. It does not end here. Why? Because there is something which is kept on top of this outer membrane as well, right? So there are these structures which are coming out of this outer membrane. What are these structures? Now these are drawn simply. When I zoom in, then I will see that at the bottom there is this structure at the bottom. Okay, this structure at the bottom. All right, no problem. Then on top of this, there is another structure like this, like so. All right. And on top of this, there is a chain. There is a chain of these polysaccharide units. Chain of polysaccharide units, right? So I cannot keep drawing so many circles. So I can just do this and as one schematic diagram, I can also make them simply to look like this. So these are nothing but zoomed in and it becomes this. What is it? This is a LPS layer. What is it? This is LPS. LPS, the lipopolysaccharide layer of the gram negative cell wall. Now the beautiful thing is that each and every component of this LPS is also LPS. All right. So when I say LPS, LPS, so I am labeling the lipopolysaccharide layer of the gram-negative cell wall. Lipopolysaccharide layer of the gram-negative cell wall. The inner to outer, as we go from inside to outside, what are all the components? The components are also LPS. So what is it? The bottommost, it is lipid A. Lipid A is the innermost. So don't you think lipid A is what is resting on the outer membrane? Yes. So something which is resting on the outer membrane is the lipid A, right? It is on the outer membrane. It is resting on the outer membrane. Then on top of it, there is the unit which is called as P. And what is P? P is nothing but core polysaccharide. Core polysaccharide. What does core polysaccharide do? Nothing. It just provides structural integrity to the cell wall. Then S, the outermost S, is the O-specific side chain, or you can also simply remember it as side chain. O-specific side chain. So you must have heard that O-specific side chain is what we use for serotyping of the bacteria, the surface typing or the serotyping of the bacteria, right? So if I ask you what are all the parts of the cell wall of a gram-negative bacteria, there is the periplasmic space, the very thin peptidoglycan, outer membrane, porin channels, and the lipopolysaccharide layer which is resting on top of the outer membrane. So after that, there is the LPS. So all of this constitutes the cell wall of the gram-negative bacteria. Now there is something very special about the lipid A. Usually you are asked to make the diagram of LPS as well. So you can very well draw the diagram and you can label the parts. The special thing about lipid A is that this is the one which ends up becoming the endotoxin of the bacteria. Yes. So if you remember, the endotoxin, endo means inside. It is a toxin which is a part of the cell wall of the bacteria. So what part of the cell wall? The lipid A part, the lipopolysaccharide layer part, and more specifically the lipid A part is what makes the endotoxin of the bacteria. Whereas exotoxins are exo, so they are like missiles which are secreted out of the bacteria, but endotoxin is very much a part of the cell wall, a part of the lipopolysaccharide layer. All right. Now, we had very simply said that here it is a very thick peptidoglycan and here is a very thin peptidoglycan. But what happens is that they ask you to draw the diagrams of this peptidoglycan layer as well. So the peptidoglycan, as the name suggests, is made up of proteins and sugar. So it has alternate repeating sugar units. You can see the green sugars and the red sugars which are repeating one after the other. What are they called as? The green one is called as NAG, and the red one is called as NAM. So NAG stands for N-acetyl glucosamine and NAM stands for N-acetyl muramic acid. Yes, so you know it's an easy way to remember is जब भी तुम जिंदगी में नाम कमाते हो तो तुम्हारे नाग जैसे दोस्त तुम्हारे साथ आते हैं, right? So wherever there is नाम, there is nag or the snakes. So you know that this is the alternating repeated units of NAM and NAG. Over here you can see this is NAG, this is NAM, this is NAG, this is NAM, this is NAG, this is NAM. Similarly here this is NAM, this is NAG, NAM, NAG, NAM and NAG. Yes. So what are these NAM and NAG doing? They are just joined alternatively, and there is something which is extending from the sides of these sugar molecules. What is that? That is the peptido part, the protein part. What is the peptido part? It's the tetrapeptide side chains. Tetrapeptide, 4 amino acids. Now, which of the two do you think is going to have an extension? So an easy way to remember is you have a name has a surname, right? So नाम का surname होता है, right? नाम का extension होता है. So which one of these two is going to have an extension? It's the name or the naam, not the nag. So extension is going to come down from this NAM molecules. And what is going to be there in the extension? There is going to be the tetrapeptide. So here I am drawing your 4 peptide molecules. right? These are the four tetrapeptide molecules which are hanging down from the NAM unit, right? So you can very easily draw this in the exam as well. So you can draw this. So from every NAM unit, they are going to come out. So here I am drawing them at almost every NAM unit I am drawing them. So even in this upper chain, it is going to be there. Even in this lower chain, it is going to be there. So let me just draw them. All right. Yes, there you go. Yes, so from almost every NAM unit, these are the extensions which are going to come out. Now, what happens very, very beautifully that in the gram positive cell wall, there is an extension of 5 hearts. So, gram positive is very, very positive. So, positive people have a very good heart, lots of heart. Yes, so these are going to have 5, P for positive, P for 5, pentaglycine bridges. Penta means 5. So there is pentaglycine bridges. So there are a set of 5 hearts, 1, 2, 3, 4, 5, which are sort of coming out and extending one tetrapeptide side chain to the other. So the third amino acid of this chain is going to connect with the fourth amino acid of this one with the help of these 5 hearts or the pentaglycine bridges. Okay, pentaglycine bridges. This is like a bridge which is connecting the third amino acid of this side chain to the fourth amino acid of this side chain. What are they called as? They are called as the pentaglycine bridges. Now, what is the name of these guys? The tetrapeptides. Very difficult. Okay, so I have a beautiful mnemonic for you. What is your mnemonic? The mnemonic goes something like Pagla, Pagla, LD, LD. Pagla, LD, LD. Rest. Just remember Pagla, LD, LD. So what is Pagla, LD, LD? P stands for gram positive. So P stands for gram positive and P stands for pentaglycine bridges. Pentaglycine bridges. Okay. What is Agla, LD, LD? Then you just draw. So the red molecule is A. Okay, first let's write LD, LD. So what were the molecules? There was the red molecule, there was the orange molecule, there was the blue one, and then there was the green one. Sorry. The blue one and then the green one. Yes, these were the molecules. So the mnemonic is Agla, LD, LD. So let me just write here A, G, La. Yes. And LD, LD. So let me write LD, LD. LD, LD. Right? So these are your tetrapeptide side chains. So A से क्या बनेगा? L alanine, D glutamine, L lysine and D alanine. Quite easy, quite simple. Yes. So these are the tetrapeptide side chains of a gram-positive cell wall. So the gram-positive peptidoglycan can be summed up with a single mnemonic, which is Pagla LD-LD. So a gram-positive has a pentaglycine bridge, and the 4 tetrapeptides, which are an extension of the NAM, are the Pagla LD-LD, which are the L-alanine, D-glutamine, L-lysine, and D-alanine. All right. And then, of course, the NAM and NAG, which are called as the N-acetyl glucosamine and N-acetyl muramic acid. वहीं पे the gram-negative cell wall also has the similar structures with the NAM and the NAG as the repeating units, but the only difference is in the tetrapeptide side chains, and there is no pentaglycine bridges. So this one is quite negative, so it will not have these hearts. You know, negative people don't have a heart. So these pentaglycine hearts or the pentaglycine bridges are absent. So there are going to be no pentaglycine bridges here. No pentaglycine bridges connecting. Also, the extensions of the tetrapeptide are going to be different. So the first unit is same. So I can simply write the first unit of Pagla LD-LD as your LA, which is the L-alanine. Okay, no problem. The second unit is also the same. So the second unit was the orange one, which was the D-glutamine. Okay, no problem. The third unit is what is going to be different here. So this is a different third unit, right? Okay, so over there I think the third unit was also, Okay, it was blue, so here it is pink. The third unit is different, it is pink. And then, of course, the last unit is very much same of Pagla LDLD was D-alanine. D-alanine. Now, this middle one is what is different. The third peptide, the third amino acid of the tetrapeptide side chain is what is different in the gram negative. What is the name of this? The name is meso-diamino pimelic acid. Pimelic acid. Yes. So if I ask you what is the difference between gram positive and gram negative, peptidoglycan, in gram negative there is no pentaglycine bridges, and the third amino acid is different, which is the meso-diamino pimelic acid. So now if overall you are supposed to write the differences between the gram positive and a negative cell wall, so you know the peptidoglycan layer is quite thick in the gram negative, where it is very, very thin in the gram negative. In the third position of the tetrapeptide side chain, this guy has what? It has the Pagla LDLD. So it has the Pagla L lysine. And over here it is the meso diamino pimelic acid. Yes, mesodiamino pimelic acid. Pentaglycine bridges are very much present in gram positive, but they are absent from your gram negative. The lipid content is very, very high in gram negative because of the lipopolysaccharide layer, whereas it is low in the gram positive layer. What about the what about the lipopolysaccharide? The LPS layer is only and only present in the gram negative, where it is totally absent from the gram positive bacteria. Teichoic acid is present on the other hand in gram positive and absent in gram negative. The outer membrane was only present in gram positive, whereas it was totally absent from the gram negative bacteria, right? So now you can make the differences table, you can write the differences table, you get an MCQ, you can solve it, you need to draw it, you can solve it. Now, so every bacteria has a cell membrane, then a cell wall, and outside the cell wall, some bacteria may or may not have something which is called as the capsule. So what is a capsule? Capsule is nothing but an organized glycocalyx. Organized का क्या मतलब होता है? Well fitted. Well fitted. So glycocalyx means what? Glycocalyx is like a slimy layer. Now suppose you're wearing a scarf or a loose clothing. Now that's hanging loose from your body. What if you're wearing a well-tailored, fitted, shiny suit, or like the armor of the knights? Yes, the people who used to go to war, they used to wear this armor. So this armor used to be very well fitted to the people. You can also remember like a well-fitted James Bond suit, right? So that is what is a capsule. Now it is an organized glycocalyx. The meaning of organized is that it is well fitted, it is tailor made, and it is specifically made for one particular bacteria, and then not all bacteria have a capsule, right? So what is this organized glycocalyx? For mostly all, it is polysaccharide in nature. So almost all capsulated microorganisms are going to have a polysaccharide capsule. There are only two exceptions to this rule. There are only two organisms which have a protein capsule, except only two guys which have a protein capsule. A protein capsule, yes. And what is this protein? It is the poly-D-glutamate. Poly-D-glutamate. So who are these guys? So just imagine, polysaccharide means carbohydrate. So you know people who are big and who go to the gym, and also their gym buddies, they always need to eat a lot of protein. So this is just a hack to remember. This is no way a logic. It's just a way to remember the things. So they need a lot of protein. So their capsule is going to be protein capsule. So who's the big guy of bacteriology? The big A, it is Bacillus anthracis. It's the biggest, most brawniest, most takatwar guy out of the bacterial kingdom, right? So of course, one of the examples here is going to be Bacillus anthracis, the one which is going to have a protein capsule. And now you know that most gym goers, they don't go alone. They go along with a gym buddy. So, you know, in Hindi, buddy is yaar. So, Bacillus anthracis ka yaar kaun hai? Yersinia, right? So, Yersinia pestis is the gym bro of Bacillus anthracis, which also has a protein capsule. Rest all are going to have your polysaccharide capsules. Even if it is fungus, even if it is bacteria, if they have a capsule, it has to be polysaccharide in nature, except Bacillus anthracis and Yersinia pestis. Now, what is a mnemonic to remember the capsules? Yikes, super killer bacteria have nice capsules. So, yikes say, of course, you have Yersinia pestis, which of course is a protein capsule. And here you have bacteria, which is Bacillus anthracis, which of course is your protein capsule. So, these two I have labeled separately. Now, let's mark the other ones. Super has S and P in its name. So, what do you remember? Streptococcus pneumoniae also has S and P. So, pneumococcus or Streptococcus pneumoniae is a capsulated bacteria with the lanceolate shape or the flame shaped. Killer is Klebsiella pneumoniae. Klebsiella pneumoniae is very much capsulated. That is why you have those mucoid colonies on the agar plate. Then have is Haemophilus influenzae. Haemophilus influenzae, how to remember? You know, I always tell you that the brain is inside a capsule. What is the capsule? Skull. So, the infections of the brain, meningitis, is also caused by the bacteria which have a capsule. So, meningitis is caused by Streptococcus pneumoniae. It is caused by Haemophilus influenzae, and which is the nice guy which causes meningitis, the Neisseria. So, your Neisseria, only and only meningitis, not gonorrhea. Not gonorrhea, only and only meningitis is what is going to have a capsule. So, not all Neisseria, only Neisseria meningitis. Then capsule is the fungus Cryptococcus. So, the only and only fungus that you need to know which has a capsule is Cryptococcus. So, now Cryptococcus capsule is also made up of polysaccharide, not just bacterial capsules. So, now you can easily remember the names of the bacteria, you can remember the nature of the capsule. Now, you have to demonstrate the capsule in the laboratory. So, of course, you can do something which is called as the negative staining or the India ink staining. What happens in the negative stain or the India ink stain or the nigrosin stain? That everything else in the background takes up the black color stain, whereas the capsule remains unstained. So, against a black background, you see shiny white capsule. Secondly, you can do something which is called as the McFadden stain. McFadden reaction or McFadden stain, in which we use polychromatic methylene blue. Polychromatic methylene blue. So, what happens? Bacteria stains blue and the capsule stains purple. This is specially done for your Bacillus anthracis bacteria. How is it going to look like? The bacteria is going to stain blue, whereas the capsule is going to take on the purple stain. So, that is the polychromatic nature. This is McFadden reaction. Then the third thing that you can do is the Quellung or the Swellung reaction. So, what happens in the Quellung reaction, which is also called as Neufeld reaction? There is a swelling of the capsule in presence of specific antitoxin. So, the capsule swells up when I put specific antitoxin on top of it. So, swelling of the capsule, and the capsule also becomes refractile. Swelling of the capsule becomes refractile on adding anticapsular antitoxin or specific antitoxin to that solution. Okay? And third, of course, is your latex agglutination test or LAT. Latex agglutination test, which can be performed directly on the patient sample, such as the CSF sample for bacterial meningitis. अब किससे होता है bacterial meningitis? It is done with it is because of strep pneumoniae, H. influenzae and Neisseria meningitis. So, for them you can do latex agglutination test, in which you see this agglutination or clump formation on a black latex card. Alright. Now, finally, you have to talk about the functions of the capsule. So, if we get a short note on capsule, you're going to write about the nature of the capsule, the examples of organisms, the demonstration, the 4 important lab diagnostic methods, and of course, the functions. Now, many students get confused in the functions of the capsule, that what are the functions of a capsule. So, an easiest way to remember it is to remember this armor of knights. What is this armor? It is well fitted, it is tailored, it was custom made for every warrior. What was the major, most important function of this armor? It was to provide protection to the person. Similarly, the capsule provides protection to the bacteria against antibiotics, against toxins, against chemicals and metabolites in the environment. So, first is of course the protection. अब जब whenever a warrior is protected from all the arrows which is coming its way, it becomes very, very dangerous. Yes, that is why he's protected from everything, so he's dangerous to others. Similarly, when a bacteria is dangerous, we call that virulence. So presence of capsule provides virulence to the bacteria. Virulence to the bacteria. If capsule is present, the bacteria becomes very, very virulent and dangerous, right? And these, if you have visited these forts and these killas, you must have seen that whenever they display these armors, every king had a specific armor. Somebody had it made it out of gold, somebody had silver, somebody was titanium, somebody was steel, other metals. So every guy had a very specific armor which was a signature of that king. So what is the signature of a bacteria? The antigens present on its surface. How does a bacteria tell you that I am this guy? It is the antigens on their surface, right? So the capsule is also what is very, very antigenic in nature. Capsule is very, very antigenic in nature. So if it is antigenic, I can definitely use it for for the diagnosis, right? So it can be used for lab diagnosis, in the lab. Yes, I can detect the capsular antigen of all of these organisms to do the lab diagnosis. My body is going to produce antibodies against this. So this is the guy which is going to be immunogenic for my body as well. Yes, immunogenic. My body will also identify it using the capsule and produce antibodies specifically against the capsule. Now, another important thing is that it has been seen that it is so important for the pathogenicity and for the survival of bacteria that sometimes whenever we remove a capsule of a bacteria, they are rendered useless. Yes, so they become so non useless that they sometimes even die if we remove the capsule of a bacteria, right? And they help so much that they also help in the formation of something which is called as the biofilm. So we are going to discuss biofilm formation today. Yes, we are going to discuss biofilm in this segment. So they also make something which is called as the biofilm. So it is the capsule which helps the bacteria make a biofilm. So these are all the important functions that are of the capsule. Now we are on the another cell appendage which is pili. You know that pili and fimbriae actually means the same goddamn thing. So pili simply means like the thick hair and fimbriae means like the thin hair. So if you just imagine all the types of hair on human body, so the body hair is very, very fine. So you can call that fimbriae. The beard hair in boys is very thick and small, or you can imagine a crew cut, that is a pili, and the long ponytail of girls, that is the flagella. So this is the way in which the bacterial cell has all of these important bacterial appendages, right? So the pili and fimbriae are very important. They actually mean the same thing. So here is a bacteria. If this is the flagella, then these small little hairs on its body is the pili or the fimbriae. They are made up of the fimbrine protein. Fimbrine protein, they mean the exact same thing. The function, what is the function of these pili and fimbriae? The function is number one, adhesion. So these are the most important adhesion molecules. Yes, pili is the most important adhesion molecules, especially for bacteria like Neisseria gonorrhoeae, which help the bacteria attach to the mucous membrane. Yes, then the second important thing, the important function of pili is conjugation. So pili, they make the sex pilus, which helps in transfer of genetic material from one bacteria to the other, right? So adhesion and conjugation are the two most important functions of the pili. Yes, so fimbriae are kind of useless, but they also help in attachment to the surfaces. But it is the pili which is the most advanced form of fimbriae, which show all of these functions. Now, demonstration. How are you going to demonstrate in the lab the presence of these pili? Now, pili are very, very thin. They are in nanometer. These proteins are very thin. They cannot be visualized with a normal microscope. So, for this, you need an electron microscope. So, either you do electron microscopy and you bring about an image, or number two, you do something which is called as a surface pellicle. So, if I have a broth in which I have a bacteria which has a pili, pili will keep the bacteria floating on the top surface itself. So this is called as surface pellicle. So I can demonstrate by the presence of surface pellicle. And number 3, I can also do heme agglutination reactions in order to prove that the bacteria has pili. So these are the 3 ways in which you can demonstrate the presence of pili or demonstrate pili inside the lab. All right, next. What is next? Now, spore. So many people, they tend to confuse capsules and spores, right? So like I always tell you, capsule is like a well fitted tailored suit of the bacteria, whereas a spore is like a overcoat. So a suit can be a part of the daily wear of any person, whereas a coat only comes out in unfavorable conditions, right? You don't wear an overcoat every day. You only wear an overcoat when you go to colder climates. So when the outside temperature is unfavorable, outside conditions are unfavorable, then the coat comes out, and it can be removed also very easily, right? So that is a spore of a bacteria. The spore is only formed under unfavorable conditions. Yes, under unfavorable conditions the spore is formed, and when the favorable conditions come back, the spore is removed by the bacteria. Yes, the spore is removed by the bacteria. So it is a multi-layer structure. So if a single bacterium, it puts on this multiple layers on top of its body in order to protect itself from unfavorable conditions. Just like when you go to extreme cold climates, you don't just put on an overcoat. What do? You first wear one layer of slacks, then you wear one layer of warmer, then you wear your shirt, and then you wear your coat, right? So similarly, the bacteria also wears all of these things. Now, the most important thing that a bacteria has to preserve, क्या होता है, whenever there is unfavorable conditions, the bacteria is not able to divide. And you know that division and multiplication is the only one survival technique of bacteria. If a bacteria cannot multiply, it dies. There is no life. Yes. Now, if the unfavorable conditions are there, bacteria needs a break. It needs to, stop for a minute. For that time, pass that unfavorable condition, and when the favorable condition comes back again, it starts dividing again. So for this period, it has to preserve its genetic material or DNA. That is the only thing that it needs to preserve. So in the center, in the very center of the spore, is your genetic material or the DNA or the chromosome of the bacteria, which has been preserved inside this many layers of the spore. ठीक है, उसके बाद after that is something which is called as the cortex. What is a cortex? It's the innermost layer. Yes, so cortex is like the warmer that you wear. Then on top of that, there is the spore coat. Sorry. Now, on top of the genetic material, there is the core. There is the core of the bacterium. Yes, core is the very center. On top of the core, there is the cortex. Now we have come on to the outermost. So core is like the skin and cortex is like the warmer that you're wearing. On top of that, you will wear your shirt or your spore coat. Spore coat, and the outermost layer is what is called as the exosporium. Exosporium means which is even outside the spore, the exo or the outermost layer of the spore. Yes, so these are your layers of the bacterial spore, and you will draw this diagram and you will label all of these structures. Now, another important thing that you have to make or write in the answer sheet is the bacteria which produce these spores. Okay, so bacteria which produce the spores, So you have to remember two guys which produce endospores. The mnemonic is B and C. B and C. So you have the Bacillus species or the Bacillus group which produces the spores, and you have the Clostridium group. Clostridium species or the Clostridium group. Now these two are the ones which produce something which is called as an endospore. Endospore. While an exospore can be produced by many people, an endospore is only produced by these two. What is an endospore? For that we have to understand the position and shape of these spores. So first of all, you have to understand that a spore can be outside or inside the body of a bacterium. If this is bacteria, this is a bacteria, the spore can be present either outside, which is called as an exospore, or the spore can be present inside the body, which is called as an endospore, right? So this is an exospore and this is an endospore. And when I talk about endospores, we have to talk about the positions. When I talk about endospore, I have to talk about the positions, different positions and shapes. So if this is a bacteria, this is a bacteria, this is a bacteria, this is a bacteria, the spore can be present at the terminal end and it can be round. So this is a round terminal spore. It can be present at the end, but it can be oval. So it is an oval terminal spore. It can be present somewhere between the end and the center. So this is subterminal. This is neither the center nor the end. Or it could be present very much in the center of the bacteria, which is the central spore. Another important thing is that this endospore, right? This endospore, so these are the 4 possible positions. These are the 4 possible positions. And another way to classify the endospore is that either they can be bulging spores or they can be non-bulging spores. Bulging spores or non-bulging spores. So what is a bulging spore? If this is a bacteria, the spore is coming out of the body. So it is almost giving it like a pregnant belly shape. Yes, so it is coming out of the body. So the bacterial shape will be somewhat like a pregnant belly. Whereas when we talk about a non-bulging spore, so if this is the bacteria, the spore is very well contained inside the body of the bacterium and there is no protrusion which is coming out. So a bulging spore gives a protrusion, whereas a non-bulging spore does not give a protrusion. So bulging spores are what are produced by a Clostridium group. Yes, bulging spores are produced by the Clostridium. So B C yaad kara tha na, so B is not B. So bulging is Clostridium, whereas Bacillus group is going to be your non-bulging. So B is not B. B is not B. So Bacillus is non-bulging, whereas Clostridium is bulging. Now when you look at the position, this terminal round spore is kind of looking like the sticks of a drummer. So this is also called as a drumstick appearance. Drumstick appearance. Whereas this guy is looking like a tennis racket. Tennis racket appearance. Now both of these are your terminal spores. One is a terminal round spore and one is a terminal oval spore. Yes, this is terminal round and this is terminal oval. So terminal round spore, which gives the drumstick appearance, is shown by Clostridium tetani. Clostridium tetani, and this is shown by Clostridium tertium. All right. So very easy to remember is T for terminal and T for tetany and tertium. Whereas tetany looks like a drumstick, so that is why it is the terminal round spore. Whereas this, the subterminal one, what is it? It is a subterminal spore. This here is subterminal because it is neither center nor terminal, so it is subterminal, and this here is a central spore right in the middle of the bacteria. So subterminal spore is shown by all the other species of Clostridium. So you have Clostridium perfringens, which shows the subterminal spores. You have Clostridium botulinum, which shows the subterminal spores. Clostridioides difficile shows the subterminal spores, whereas the central spore is by Clostridioides, by Clostridium bifringens. So bifringens is fermenting bifurcating into two. So the middle spore is bifurcating the bacteria into two equal halves. That's why. But that is not very medically important. But just to write an example, you can write that as well. So this is the examples of position and shape of the spore. Now you have to talk about the sporulation cycle. So I've already drawn it for you, but you have to draw all of this in the diagram in the exam. What happens? You have this vegetative cell. Vegetative cell simply means a normal bacteria. Now, whenever there is unfavorable conditions, the bacteria has to start producing the spore. So, how does it produce a spore? First of all, the first thing which happens is that the inner wall develops something which is like this septations. So, these septations are developed inside the wall. What is the first step? It is the septations. Septations which are formed. Then the septation divides the inner cell into two parts. Yes. So this one is called as a forespore. So you can see that the genetic material is also divided. And this one is called as the germinal cell or the mother cell. The germinal or the mother cell and the forespore. Why is it a forespore? Because this is the guy which is going to become a spore in the future. The next step which happens is that the mother cell engulfs the forespore. There is an engulfment. So the mother cell engulfs the forespore. Engulfs the forespore. Now the mother cell's DNA is almost lost. What is remaining simply is the forespore which has become like a complete cell with its DNA, right? So there is loss of maternal DNA. Loss of maternal DNA or mother cell DNA, and finally you have the deposition of the other layers of the spore. So you can see over here there is the chromosome, the green cell wall, then the orange and the red layers have been added. So what are these orange and red layers which have been added? So you have the cortex and the spore coat. The cortex and the spore coat gets laid, deposited, okay, gets deposited. Finally you have this totally mature spore which is released out of the bacterial cell. Now whenever the favorable conditions come back, the bacteria has to come out of this spore. So you see that all the layers have opened up and the bacterial cell has germinated, and after germination the vegetative cell is back. So the entire cycle of sporulation is complete, and you must draw this in the exam as well. Now of course we have seen the position and shapes and examples. Now we have to talk about the demonstration of the spores. Now staining is the mainstay. Staining is the mainstay of demonstration of spores. So when I put a particular stain, I see the spore taking another color and the bacteria taking some other color. So the two different stains which are taken. So I can do the simplest of all, which is the gram stain. What will the gram stain do? In the gram stain, the bacteria takes the gram stain, but the spore remains unstained. Yes, the spore remains unstained. So I can see empty spaces. Suppose I have a gram positive bacilli. I have this gram positive bacilli, yes, and when the spore will not take up the stain, so I am going to see these empty spaces inside the cell. These empty spaces are going to tell me that this has a spore. Yes, the second important method is called as the Schaefer-Fulton method. Schaefer-Fulton method. In this I use two dyes. I use malachite green and safranin. So my bacteria turns green because of malachite green, and the spores take up the red stain because of the safranin. Yes. This is the Schaefer-Fulton method, in which I am using your malachite green and your safranin. The two stains which I use are malachite green and safranin. Then we have another method which is called as the Moller's method. Moller's method. In the Moller's method, you have the blue colored bacteria and the red colored spores. I'm sorry, in Schaefer-Fulton, you have the green colored spores. The malachite is what is taking, malachite green is what is staining the spores. Okay, all right. I mean, it's not that important. Just remember that gram stain, the spore remains unstained. In Schaefer-Fulton, we are using malachite green and safranin. And in Moller's method, we have the safranin and the methylene blue, in which we have the blue and the red color production. So these are the mainstay of demonstration of spores, where I am using different methods of staining. Now, a very very important topic is toxins. Toxins. तो अभी हमने bacterial cell wall में देखा था that endotoxins are a part of the cell wall of the bacteria which are present in the lipopolysaccharide layer, whereas exotoxins are what are released out of the cell. So if this is the way to remember, and all my students would know that we always remember exotoxin and endotoxin based on these two images. So over here you can see the blue colored avatar people who are throwing arrows. So you can remember exotoxin as an arrow which is thrown by the bacteria, or maybe an intelligent missile with all the cameras and the specificity. It goes and falls on a specific tissue target. Similarly, these arrows which are released by the avatar people, they are very tiny arrows, but they are very very fatal. A small arrow can take down a whole horse, and that is also very specific. So all of these are the features of an exotoxin. So first of all, these are the avatar people, which are the blue people. So exotoxins are released by your gram-positive bacteria. They are released by the gram-positive bacteria. What is the nature? Nature, P for positive, P for protein. So these are protein in nature. Yes, what is the location? They are excreted out. They are secreted out of the cell. There is no location. They are not present inside the bacterial cell wall. They are secreted out of the cell. Yes, and they are released by secretion, or they are, sent out of the cell. Heat stability. All of these intelligent missiles and all, if you put heat to him, they will go bad. So these are heat labile. These are not heat stable. What about specificity? Like I said, they are highly specific. They are highly specific. So they will only go and hit one specific tissue target. That is why you will see that examples of exotoxin are like neurotoxin and enterotoxin. So they are not going to get lost in the way. They will go and hit exactly the nerve that they are supposed to hit. Fatal dose. So what are these? These are tiny arrows which can take down a horse. So even a small amount of exotoxin is enough to kill an organism. That is why they are fatal in small doses. Fatal even in small doses. And antigenicity. If something which is so fatal, it is going to be highly antigenic, and something which is highly antigenic is also going to be highly immunogenic. So if I give this antigenic thing inside my body as a vaccine, my body will produce a lot of immune response, and that is what I want to achieve by giving a vaccine. That is why vaccines in the form of toxoid vaccines are available against the exotoxins. So bacteria which produce exotoxins, you can give toxoid vaccines against them. Example, diphtheria toxin, tetanus toxin, you give the toxoid vaccine. So these are what? These are exotoxins released by the bacteria, but vaccine prevention is possible. These guys are coded for by the plasmids. So don't you think that two bacteria can exchange plasmids? So the property to produce exotoxin can be exchanged between two different bacteria. Whereas on the other hand, in endotoxins, you can first of all see that it has the letter N in it. So it is obviously going to be secreted majorly by the gram negatives. Another way to remember is this guy. He's a suicide bomber who's wearing a red t-shirt. So it's easy for you to remember. What is the property of a suicide bomber? They strap bombs to their body. Now, if they are supposed to kill somebody, they have to die themselves. Yes, they burst and they die themselves. That is the property of your endotoxins. So endotoxins are definitely released by the gram-negative bacteria, and they are present in the lipopolysaccharide. So the nature is lipopolysaccharide. They are present in the cell wall of the bacteria in the lipopolysaccharide layer. What was the layer? They were lipid A. I had told you that lipid A is what is the endotoxin of a bacteria. Yes, I told you that lipid A is what is the endotoxin of a bacteria. So what is the nature? It is the lipid A. Where are they present? They are present in the lipopolysaccharide layer of the cell wall. How are they released? This is very important. Now, in order to release an endotoxin, the bacteria has to lyse itself, right? So there is lysis of bacterial cell in order to release the toxin. So the bacterial cell has to sacrifice itself. It has to kill itself in order to kill the others with the help of endotoxins. They are very much heat stable. So heat से इनका कुछ नहीं होता है, and they are not very specific. They are not tissue target specific. They are not tissue target specific. So they will lead to conditions like shock, wherein the entire body takes a toll. So they are like bombs which burst inside the body. So when one bomb bursts, everything around gets damaged. So there is high tissue destruction and shock like status, but there is no tissue target specificity which is there. They are fatal in high doses. They are fatal in high doses. You need lots and lots of endotoxin to be released before it can kill a person. And the antigenicity is also low. That is why you have no vaccines against these endotoxins. There are no vaccines which exist against these endotoxins. Now, they are a part of the cell wall. One bacteria cannot give its cell wall to the other bacteria. That's not possible. But who can they give it to? A mother can pass on that trait to a child. So they are chromosome coded and they can only be transmitted from mother to child. So endotoxins का सिर्फ vertical transmission possible है, and exotoxins can be exchanged between two bacteria sitting next to each other because they can transfer plasmid to each other. So this finishes all the important differences between an exotoxin and an endotoxin, which can be very easily remembered with a simple image of the Avatar people shooting arrows and the suicide bomber wearing a red t-shirt. Yes, and of course the N helps you remember that it is released by the endotoxins. Now, in the physiology of bacteria, we have one very, very important concept, which is the bacterial growth curve. Yes, तो bacterial growth curve आसान है बहुत. You might get MCQs related to it. You might get a short note related to it. Mostly what they ask is the MCQs. So we always draw bacterial growth curve like this. And we all know that initially, first of all, there is a lag phase which happens in which there is no bacterial cell division. So no division takes place, no increase in the cell number happens. So what are we plotting? On the x-axis, we are plotting the time, and on the y-axis, we are plotting the number of cells or cell division. So if cell division will happen, graph will rise up. If no cell division is happening, graph will be flat. So this is the lag phase where the graph is flat. Then you have the log phase in which there is exponential cell division. So you can see that in a very, very short amount of time, the number of bacteria has increased exponentially. So that is the log phase wherein there is extreme cell division and very, very high number of bacteria are produced. Then finally, you have the stationary phase. What happens in stationary phase? That the bacteria now start dying. They start dying, and therefore the number of new bacteria which is being multiplied and the number of deaths is equal. So that is why overall there is no increase in population. And finally, in the end, there is something which is called as the death or the decline phase, in which there is decline or death in the number of cells, because now the death is much more than the number of cell divisions. So if you have to label it correctly, you will say here there is no cell division. No cell division, only increase in size. So bacteria is just sitting and eating nicely. बैठ के आराम से खा रहा है, and it is just getting fatter and fatter. What happens in the log phase? There is exponential cell division, exponential multiplication and exponential cell division which happens. What happens in stationary phase? Cell division has become equal to cell death. So the number of cells dying is equal to the new cells which are getting produced. That is why there is no shift. And whereas in decline, there is a lot more cell death which happens as compared to cell division. So overall, the population starts declining. But this is the curve which is shown by the viable bacteria or the living bacteria. Yes, whereas the total count means the total body count, which is living plus dead. So over here, even though the cell division is equal to cell death, but the total number of cells inside the test tube is still increasing. Yes. So if I have to draw the total count, how will I draw it? Of course, in total count also, there is going to be a lag phase, then there is going to be an exponential log phase, but the stationary phase will not be stationary. that number is going to keep increasing a little bit. So the total count keeps on increasing and there is no decline. Why? Because total count means live plus dead. What have I done? I have taken a test tube and in that test tube I have added 10 bacteria. ठीक है, कैसे मैं करती हूँ growth curve? How do I bring about a growth curve? I've taken a test tube in which I add 10 bacteria and then there is nutrition in it. First of all, those 10 bacteria are not dividing, they are simply sitting and eating and becoming fat. So the number remains 10. Time is passing, but number remains 10. There is no increase in number. Finally, these 10 bacteria, they start multiplying exponentially. 10 ka hua 20, 20 ka 40, 40 ka 80, 80 ka 160. So exponentially, they are dividing. And within a small amount of time, they have increased a lot. All of these are alive, viable, live bacteria that we're talking about. Now, a time will come when nutrition will start depleting, because in that test tube the amount of broth that I had added was fixed, but the bacteria is multiplying and population is increasing. So now nutrition will start falling, so death will start to occur. Over here the cell division and the number of cell death is equal. So if 10 bacteria are dying, 10 new are getting produced. So but those cells which are dying are also inside the test tube only. Na, ऐसा थोड़ी है मर के bacteria will leave the test tube and go outside. So inside the test tube, if I count the total number of cells, living as well as dead, that is continuously increasing, because over here cell division has still not stopped. Cell division is still happening. It's just that equal death is happening. So when I count the number of alive cells, that does not change. But when I count the number of total cells alive as well as dead, that keeps on increasing. So the blue line, dotted line is giving me the total count which keeps on increasing, and this whole shebang which is there with the shapes, this is simply giving me the viable count of the bacteria. Viable count, yes. So maximum viable count is achieved at the end of the log phase. Yes, after log phase, the viable count is not increasing. But maximum viable count is achieved at the end of stationary phase. Over here, total count maximum is at the end of stationary phase. So this is the MCQ that they ask you. When is the maximum size of bacteria achieved? End of lag phase. When is maximum number of live cells achieved? End of log phase. When is the maximum number of total cells achieved? End of stationary phase. Yes, stationary phase. Now we have to talk about the log phase and the stationary phase. Before that, let's talk about decline and let's get it over with. Just to sum up in a single word, while lag is like a couch potato where the bacteria is just sitting and eating and becoming fat, log is equal to jog. So log phase is like the youth of bacteria. Yes, log is jog, meaning it is active, it is the youth. And finally, stationary phase is like a war zone. It's like a war zone. So what happens after war? There is mutilated bodies. So what are the mutilated bodies of bacteria? Called as involution forms. So what happens in the decline phase is that bacteria makes involution forms or the abnormal forms. So you know, you can write about lag phase, you can write about decline phase. Now you just have to write details of log phase and stationary phase. So let me just write it here. What was log phase? It was the youth of bacteria. It was the jog phase. Yes. So what happens when you're young? You are very, very active. So you are metabolically active. That means you are very, very active in this phase. The bacteria is also biochemically active. What is the meaning? When you are young, you want to talk to so many people. You want to go and interact with so many people. That is biochemical activity. The bacteria is going and talking to many, many people. That is biochemically most active. At this time, you are very, very sensitive. Yes. So if someone calls you something, you feel very bad very fast. So this is the phase in which the bacteria is most susceptible to antibiotics. So if I tell you what is the best time to perform biochemical test and antimicrobial susceptibility, of course, when the bacteria is in the log phase, it is the best time to perform the biochemical tests plus the antimicrobial susceptibility test. Yes. Then when you are young, you care about makeup and your looks. So at this time, the gram stain is going to be uniformly stained. Yes, it's going to love makeup. It is going to love gram stain. So the best time to perform gram stain is your log phase. Yes, best time to perform gram stain is log phase. Best time to perform gram stain is log phase. Then finally, what happens? What else happens? This is the time when you care about shiny cars, tailored suits. Yes. So what is going to get formed? Capsule. Many people confuse this capsule formation and spore formation. While S for spore formation happens in S for stationary phase, the capsule formation happens in the log phase, because this is the stage of life where you care about tailored suits and shiny cars. So just imagine capsule as a shiny car or a well-fitted tailored suit and you're good to go, right? Then finally, what happens in stationary phase? Like I said, stationary phase is like a war zone. Stationary phase is like a war zone. So what happens in a war zone? In a war zone, there are two types of people. One which go and hide and one which put up a fight. So the hiders will go and hide inside bunkers. They will hide inside blankets. So what is that? Spore. Hiders will take boxes full of food so that they can sustain for months underground. So what is that? Storage granules. So S for stationary phase, S for sporulation, and S for storage granules. So if I ask you, when is the spore formed? Stationary phase. When is the capsule formed? Log phase. Yes. When are the storage granules, also called as the volutin granules of Corynebacterium diphtheria, formed? They are formed in the stationary phase. Then you have the fighters, the guys which put up a fight. So the guys who put up a fight, how are they going to fight? They're going to fight with missiles and arrows and guns. Yes. So what are these missiles and arrows and guns and bombs? The suicide bomb and the missiles, the toxins. So all the toxins are produced in this phase. Both endotoxin as well as exotoxin are produced in the stationary phase by the bacteria. Why is bacteria producing toxin? क्योंकि अब न्यूट्रिशनल डेफिशिएंसी है. वॉर क्यों होते हैं? जब रिसोर्सेज खत्म हो जाते हैं. वॉर्स हैपन व्हेन देयर इज नो रिसोर्सेज. Now inside that test tube, the resources are depleting, the broth is depleting. So now the bacteria has to put up a fight. It has to kill others to survive, right? So it will produce these toxins. This is the time in which antibiotics are produced by the bacteria. Now please don't confuse it. In the log phase, what was happening was the bacteria was susceptible to antibiotics. So if I wanted to see whether one antibiotic will kill a bacteria, log phase was the best time. But stationary phase is when the bacteria starts producing antibiotics to kill the other bacteria. So you know that antibiotics are in fact derived from bacteria itself. Along with antibiotics, the bacteria also produces bacteriocins. So bacteriocins, toxins, antibiotics are all produced by the bacteria in the stationary phase because it wants to survive and it has now become offensive to the other bacteria inside the test tube. At this time in the war zone, if I give you a red jacket or some makeup, would you care? You would not care about makeup in a war zone. That is why in this phase, the gram stain is totally non-uniform. Yes, so this is not a good type to do a gram stain. The bacteria is non-uniformly gram stained during the stationary phase. So all of the points about stationary phase are done, all of the points about log phase are done, and all of the points about decline phase as well as lag phase are done. You can make two types of curves. One is the viable count curve and one is the total count curve, which are totally different whenever we talk about the bacterial growth curve. All right. So that's it in the bacterial growth curve. After that, when we discuss the physiology of bacteria, one very, very important point is the methods of anaerobiasis. So what is anaerobiasis? It is achieving anaerobic environment. Some environment which is totally rid of oxygen. Now, how do I remove oxygen from air? What I can do it, I can control the environment inside small little setups. So I can take a jar and change the environment inside the jar. I can take a test tube and change the environment inside the test tube. I can remove all the oxygen from there. That is called as anaerobiasis. Now, while I will discuss the methods of anaerobiasis with you, there is something which is very, very important, which is called as the indicators of anaerobiasis. Now, suppose I have a jar, suppose I have a test tube. By some method, I have managed to achieve anaerobiasis inside it, meaning I've managed to extract all the oxygen out of it. How do I make sure that in fact anaerobiasis was achieved inside this and I did not fail? I will put some biological as well as some chemical indicators. So there is a chemical dye which only turns blue in the presence of oxygen, and that is methylene blue. So if my methylene blue has turned red, that is telling me that I have failed. Has turned blue, that is telling me that I have failed, because methylene blue is only blue in the presence of oxygen. So if oxygen is there, then it turns blue, and that is what is telling me that anaerobiasis has failed. But if there is no oxygen, methylene blue remains colorless, and colorless, it remaining colorless will tell me that I have passed. I have in fact maintained and successfully achieved anaerobic environment inside my test tube or inside my jar. Then the other method is called as the biological indicator. Biological indicator. If I have succeeded to achieve perfect anaerobic conditions inside the jar, obligate aerobic bacteria should not be able to grow, because obligate aerobic bacteria need 21% oxygen, right? So if at all an aerobic bacteria is in fact growing inside that test tube or that jar, that means anaerobic environment was not there, some oxygen was coming from somewhere, right? So who is that guy? Who's the prototype guy of aerobicity? It is Pseudomonas aeruginosa. Name itself says that it is aeruginosa, it loves air. So if aeruginosa grows, if Pseudomonas aeruginosa grows, that means I have failed. From somewhere oxygen has entered inside the test tube. If Pseudomonas aeruginosa doesn't grow, there is no growth, that is telling me that I have in fact passed, because now that means total perfect anaerobiasis was achieved inside my system. So what are the methods of anaerobiasis which I can perform? So one is a chemical method. Then you have the physical methods. So let's discuss the chemical method, which is the use of Robertson cooked meat broth, or the RCMB, right? This is the RCMB. This is the RCMB, or the Robertson cooked meat broth. What happens? There is meat which is cooked and minced and used inside this broth, right? So let me write the full form. It is Robertson cooked meat broth. All right, Robertson cooked meat broth or Robertson cooked meat medium. What all is there inside this medium? Inside this, there is BHI broth, brain heart infusion broth. Yes, and there is the minced plus cooked meat particles. So what are the use of both? Brain heart infusion is the bada heart and the chota brain. So you have the beef heart and the calf brain, and mixed meat or cooked meat is of the ox or the goat heart. Ox or the goat heart is what is used for the meat inside it. Why is it cooked and minced? Because cooking and mincing is going to lead to release of lots of reducing agents. So the property, the reason by which these chemicals, they help me achieve anaerobic environments, is these reducing agents. What do these reducing agents do? They reduce the redox potential. So what is the principle of using chemicals? They reduce the redox potential. Now, so research has shown that simply removing oxygen is not going to make the cut. So it is very much important to also reduce the redox potential inside the test tube to get perfect anaerobiasis. So these reducing agents are going to reduce the redox potential. And what are these reducing agents? It's the sulfahydryl groups. Sulfahydryl groups and glutathione. Those of you who are into beauty, they know that now glutathione these days is very big in youthful appearance of people. So what is aging? Aging is oxidation. So something which is an antioxidant prevents aging. So glutathione is an antioxidant. Something which is an antioxidant is a reducing agent. So even in the Robertson cooked meat broth, the meat which is the goat heart is providing these sulfahydryl groups or the glutathione, which is helping me create this reduced redox potential inside my test tube. Out of the physical methods which can be used, you have the oxygen absorption and so the second is of course the physical methods. Physical methods. So either you can do evacuation and replacement. What is evacuation and replacement? You are evacuating the entire air from a jar. So what is the jar I am using? This jar over here which has all of these Petri plates put inside. The jar that I am using is the Macintosh Fields jar. Macintosh Fields jar. And I connect this Macintosh Fields jar with a machine called as the anoxomat. So what is happening? We are evacuating all the air from inside this jar and we are replacing that air with anaerobic air. So एक jar में से मैंने सारी हवा बाहर निकाली. Now there is vacuum inside. Now from another pipe I will put in anaerobic air as per my liking. However amount of hydrogen, nitrogen, carbon dioxide I want to put, I will put inside this jar. Now this jar has perfect anaerobic environment inside. So we are creating small micro environments inside these jars for anaerobic bacteria to grow. Another method of physical methods is your oxygen absorption. So I will use some chemicals which are totally going to absorb the oxygen and remove it from this jar. One example of that is the gas pack. So what is a gas pack? It is like a small packet which has sodium bicarbonate and sodium borohydrate. This sodium bicarbonate and sodium borohydrate is going to react with the release the carbon dioxide and hydrogen, which is going to absorb all of the oxygen from inside this chamber, and it is actually oxygen absorption. Who leads to this? It is the palladium catalyst. It is very important to know that the palladium catalyst which is put inside this jar is what is catalyzing the utilization of oxygen into carbon dioxide and hydrogen. Therefore, there is no oxygen left inside this jar. So, two important physical methods are evacuation, replacement and absorption of oxygen, and one important chemical method is the reduced redox potential using the RCMB, right? So, you will write all of this. Now, coming on to antimicrobial susceptibility testing, very important. So, you have three major methods of antimicrobial susceptibility testing. You have the conventional methods, the automated methods and the molecular methods, right? So, what is conventional methods? You do phenotypic detection. What is phenotypic? It is the way the bacteria behaves with different antibiotics. So, I will take a specific bacteria, I will take a specific antibiotic and I will make the two interact and make the two try to behave with each other. Yes. So, what are the conventional phenotypic methods which are used? Either I can use a broth dilution method, or I can use a disc diffusion method, or I can use something which is called as the E-test, right? So, all of these are important: broth dilution, disc diffusion and E-test. So, we are going to discuss these in detail. Then amongst the automated methods, you have like the Vitex systems, which are automatic machines which tell me the drug-bug combinations and they tell me which is susceptible to what. And then the molecular method is basically the genotypic method, wherein I am detecting the resistance gene. Now, the problem with genotypic method is presence of gene does not mean resistance. Suppose a bacteria has a gene. What if the gene is not getting expressed? So, phenotypic methods are confirmatory, but genotypic methods, they tell me whether the gene is present or not. Example, if MEK gene is present inside Staphylococcus aureus, you know that that is an MRSA or methicillin resistant Staph aureus. So, you are detecting the genes in the molecular methods. Now, very important to know are the conventional methods. Amongst the conventional methods, we start talking with the disc diffusion. So, what are the prerequisites of disc diffusion method? What I use is the cation adjusted Muller-Hinton agar. Cation adjusted Muller-Hinton agar is what is used. I use 6 micrometer filter paper strips soaked in antibiotics. filter paper discs soaked in antibiotics. Soaked in the antibiotics of my choice. Secondly, I do a lawn culture or a carpet culture. Lawn culture or carpet culture using 0.5 McFarland standard of inoculum of bacteria. Yes, so what do I do in disc diffusion? I basically take this plate of Muller-Hinton agar, and on this plate of Muller-Hinton agar, what have I done? I have lawn cultured. So I will take my bacteria and I will culture it all over this plate like a lawn or like a carpet. Then I will take some filter paper discs which are coated and soaked with the antibiotic and I will put them across. If my bacteria is susceptible to the antibiotic, it is going to show me a zone of clearing around this disc. Yes, so what have I done? I have taken these filter paper soaked discs which have the antibiotics. If my bacteria is susceptible, there is going to be a zone of clearing around these. So I will see as if somebody has erased all of the colonies from around. So this big zones of susceptibility I am going to see around these discs. And if the bacteria is resistant, I will not see any zone. So I will label that this guy over here is susceptible, whereas this guy over here is resistant, because bacteria is going right up to the mouth of the disc. So the reason for not showing you an image and drawing this diagram is to just to make you understand that it is very much possible to draw these diagrams in the exams, right? Now you have two types of disc diffusion methods, right? So you have two types of disc diffusion methods. What are the two types of disc diffusion methods? One is called as the Kirby-Bohr's method. Kirby-Bohr method. And one is called as the Stokes method. Stokes method. So what are we doing in Kirby-Bohr method? So let's just draw the two. What is this? This here is the Kirby-Bohr method. Over here you have the lawn culture, and over here also you have the lawn culture. So both of these are cation adjusted Muller-Hinton agar, and you have the lawn culture of bacteria in both of them. What happens in the Stokes method is, in the Kirby-Bohr, is that on a single plate you apply around 5 to 6 discs. Yes, 5 to 6 discs are applied around the plate, and in this you measure the zone diameter. So around these antibiotic discs you will get the zone of inhibition. The zone of inhibition that you get, you will measure the diameter of this zone of inhibition. Yes. So what happens in Stokes method? Sorry, in the Kirby-Bohr method, in the Kirby-Bohr you measure the zone diameter. You measure the zone of inhibition diameter. Zone of inhibition diameter. So from here till here, this diameter you will measure in millimeters, and you will compare it with the CLSI charts. You will compare it with the CLSI charts, and you will know for each drug-bug combination. Yes, what happens in the Stokes method is that you divide the culture plate into two parts. Over here, you have put a culture of a known positive strain. This is a known positive strain, and this here is the test organism that you want to test. So what is this? This is a known strain. I know how this bacteria behaves with the antibiotic that I am putting, and this here is the test strain. This is the strain of bacteria which I am using on the patient. Over here, we were only using the test strain. We were only using the test strain, and we were using multiple antibiotics. So what can you do in Kirby-Bohr's? You can use one bacteria and you can use multiple antibiotics. But what happens in Stokes method is you can use a maximum of 2 antibiotics. You can use a maximum of 2 antibiotics, but you can use multiple strains of bacteria which can be tested against a known positive strain. So I know for a fact, suppose I know for a fact that my test bacterium is very much sensitive to antibiotic A, but it is resistant to antibiotic B. Now compared to my test strain, compared to my known strain, if the zone of diameter is smaller for the test strain, I know that this is resistant. But if the zone of diameter is larger for my test strain, then I know that this is sensitive. So how will I interpret this? Compared to the control or the known strain, this zone is smaller, so this here is resistant. But this zone is larger as compared to this zone, so this is very much sensitive or susceptible, right? So this is how we will draw the diagrams in the answer sheet also, and the two methods of disk diffusion is what we know. Then we have the broth dilution methods. Broth dilution. In broth dilution, there can be macro broth dilution and there can be micro broth dilution. Yes, there can be macro broth dilution and micro broth dilution. The macro broth dilution is done in test tubes. The micro is done in the micro titer plate. The only thing which changes is the amount of inoculum is in microliters. Rest everything remains the same. So what will I do? I will take many test tubes. Yes, so I either many test tubes or many small little wells of the micro titer plate. So it only changes depending upon the amount of fluid that I am using. In all of these, I have put a fixed amount of normal saline. ठीक है, suppose सब में मैंने डाला 5 ml का normal saline मैंने सब में add कर दिया है. All right. Now, I have added a fixed inoculum of bacteria. What have I added? In all of them, I have a fixed inoculum of bacteria. In all. In all test tubes, right? Now I will keep changing the concentration of the antibiotic that I am using. Suppose in this the antibiotic concentration that I am using, so here I am writing the antibiotic concentration. So antibiotic concentration in this test tube suppose is 2 micrograms. In this I have made 4 micrograms. इसमें 8 हो गया, इसमें 16 हो गया, इसमें 32 हो गया. So either what I can do is I can take 32 in this and keep on diluting, giving me serial dilutions of antibiotic, or I can keep increasing the concentration. It is more difficult to add 2 micrograms of antibiotic. It is easier to add 32 micrograms. So I can add 32 micrograms and then I can keep serially diluting the broths in order to achieve the perfect amount of dilutions of antibiotic. So what are we doing? As we are moving from here to here, we are achieving serial dilutions of antibiotics. Serial dilution of antibiotics, which is of course the doubling dilution. It is getting halved. Yes, so if the concentration is getting halved each time, the dilution is getting doubled. है ना? Concentration and dilution are equal and opposite. So this is the serial dilution of antibiotics, which is also called as the doubling dilution. Doubling dilution. और सबका जो इनोक्यूलम है यानी जो बैक्टीरिया जितना रखा है वो मैंने रखा है सेम. नाउ व्हाट विल हैपन इज सपोज द बैक्टीरिया वाज नॉट स्केयर्ड ऑफ 2 micrograms. So over here you will see lots of turbidity and you will see lots of growth. Now it was not scared of 4 microgram also. So here also you started seeing lots of turbidity. Now the turbidity started reducing a bit from 8 micrograms, but still it was visible. And finally what happened in the 16 and 32 microgram test tubes is that you could barely see any growth. So there was clearing of the broth. The broth started becoming absolutely clear. So what happened over here? In 16 microgram onwards, you did not see any visible growth. The turbidity was lost. So what is this? This here is the minimum inhibitory concentration or the MIC of that particular test. So what is MIC? MIC stands for minimum inhibitory concentration. concentration of antibiotic. What does it tell me? It is that point at which no visible growth is possible. So what is visible growth? It is turbidity. So the test tube in which I stopped seeing turbidity, that concentration was the minimum inhibitory concentration of antibiotic. So it is the minimum concentration of antibiotic or the maximum dilution of antibiotic, both mean the same thing. So the minimum concentration or maximum dilution of antibiotic needed to inhibit the visible growth means the turbidity, the visible growth of a fixed amount of bacterial inoculum. ठीक है, that is minimum inhibitory concentration. In contrast to MIC, you have something which is called as MBC. What is it? It is minimum bactericidal concentration. Minimum bactericidal concentration. What is it? It is the minimum concentration needed to absolutely kill the bacteria. So there are bacteriostatic and bactericidal drugs. So this drug is going to absolutely kill. It is the minimum concentration or maximum dilution of antibiotic needed to kill the bacteria. To kill the bacteria. So how is it seen? MIC is seen as loss of turbidity in test tube. Loss of turbidity or clearing inside the test tube. Whereas MBC is seen as, it is seen as loss of growth upon subculture. So यहाँ पे क्या हुआ था? In this test tube, ये 16 माइक्रोग्राम वाली पे मुझे turbidity दिखनी बंद हो गई थी. But when I took this broth and plated onto a culture plate, I could still see some bacteria. So then I had to move to the next concentration. So suppose at 32 micrograms, when I finally subcultured from this test tube, I did not see any colonies. So my MIC is 16, but my MBC becomes 32, right? So MBC is absolute killing, wherein there is no single colony also, even on further subculture from that test tube. So this is microbroth dilution. What happens in E-test? E-test is the best of both worlds. So I will again take this plate and I will do a lawn culture. So disc diffusion is easy to perform, but broth dilution is much more sensitive and confirmatory. But it is very cumbersome to perform and very difficult to perform. So I will not go through the pains of putting the broth dilution, but I also want, good results. So what will I do? I will take a filter paper. This filter paper is soaked with antibiotics in a gradient, right? So what was happening in the discs? In the discs, there was a fixed amount of antibiotic. What was happening in the discs? In the disc, you had a fixed amount of antibiotic. So suppose one disc has around 3 microliters of antibiotic. So that's it. But what happens in this strip? In this strip, you can put gradients. So up till here, I can soak 2 micrograms, up till here 4, then 8, 16, 32, 64. So I can keep on increasing or decreasing the concentration of antibiotic on this filter paper strip. So what is this? This is a strip soaked in gradient. So it has a gradient of antibiotics. Gradient of antibiotics. Yes. Now the zone of inhibition. How will the zone of inhibition form? Suppose the bacteria was very much scared of 62 microliters. So of course there will be a huge zone of inhibition around it. As I will move towards lower concentration, the bacteria will not be much scared. Yes. So the zone of inhibition is going to not be there. So I will get something which looks like this. Yes, so we get this kind of oval or elliptical zone of inhibition around the disc. What is it telling me? That when was the point that the bacteria started getting scared of the antibiotic? Around this point. Yes, around this point the bacteria started getting scared. इससे पहले तो bacteria was not scared. It was right up to the mouth of the antibiotic. But from here it started getting scared, and then it was really scared because the zone of inhibition kept increasing as my concentration of antibiotic was increasing down the strip. Yes. So what is this zone? It is the elliptical zone. Elliptical zone of inhibition. And what is this point at which it started telling me? This point is giving me the MIC value. So what is it? It is like best of both worlds. It is like disc diffusion but giving me the MIC. So what is this test called as? This test is called as E-test. 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 So it is best of both worlds. It is MIC by diffusion. MIC by diffusion. So अब तक हमने देखा था MIC मिलती है डाइल्यूशन से, बट डिफ्यूजन से MIC हम कैसे देखते हैं? By using the E-test. Okay, so that is the E-test, and that is how you can draw the diagrams in the exam as well. So we have seen all the important types of your disc diffusions. Now, amongst bacterial genetics, I will be talking about plasmids, then I will be quickly talking about AMR, and I expect and I hope that you will go home and you will revise your transformation, translation, transduction and conjugation. Otherwise, you can go and watch it in the one-shot series where I have covered. I have also taken separate lectures on bacterial genetics. Otherwise, the BDS FARRE General Microbiology, over there I have covered bacterial genetics, the transduction, conjugation and transformation, right? So now let's quickly talk about plasmids, because these are the topics which get ignored mostly, right? You all pay attention to the other aspects, but these are the topics which get ignored, but they are asked in the exams. So what are plasmids? So if you look at a bacteria has a nucleus. So this is the main chromosome, and this is the main chromosome or the nucleus of the bacteria. Other than this main chromosome, there are some small little genetic material which are extra chromosomal, which are freely independent, floating inside the bacterial cell. That is what is plasmid. Plasmid is extra chromosomal genetic material. So it is very much genetic material, and it is very much has all the genes, but it is extra chromosomal. It is freely floating. It is free and independent of the main chromosome. So whenever the bacterial cell is dividing, or when the bacterial chromosome is dividing, plasmid does not care. Sometimes the bacterial chromosome may not be dividing, but plasmid can divide. Plasmid can replicate. Plasmid can transfer. Plasmid is independent. It is free and independent of chromosome. So it is capable of independent replication. It causes independent replication, and it also can cause independent transfer to the other bacteria. The number is a lot. It is present in high numbers, more than 40 per cell. A single bacteria can have more than 40 plasmids inside a single cell. Yes, and there are the different types of plasmids. So you have to write about the plasmids. Yes, but they are 40, but they have a small number of genetic material, right? So one plasmid will have around 10 base pairs. So they are very, very low molecular weight. Very, very low molecular weight. Yes. Now, like I said, you have to know about the different types of plasmids. So, plasmids example can be the R plasmid. What does R plasmid do? R plasmid transfers the resistance gene between two sitting bacteria. The thing about plasmids is that they can be transferred between two unrelated bacteria. So, two bacteria sitting in close proximity can transfer and exchange their plasmids, whereas chromosome can only be exchanged between mother to child. Chromosome can only be passed from in vertical from mother to child. But okay, 5 minutes. कितने बजे से है नेक्स्ट क्लास? 12 बजे से. हाँ, ठीक है. Yeah, this independent transfer is that the plasmids can be exchanged between two unrelated bacteria. between two unrelated bacteria, whereas chromosome is only transferred vertically from mother to child. Yes. So like I said, R plasmid is the one which is going to transfer the resistance gene. Then there is the fertility plasmid or the F plasmid, which transfers the ability to make the sex pilus. Yes, it is responsible for fertility, for exchange of for the production of sex pilus. Then you have the col plasmids, which make it possible for the bacteria to exchange the colicin producing capabilities. Then of course you have the virulence plasmids, which help in exchange of virulence. So if I am a very virulent bacteria, I can make you virulent. Then of course there is the metabolic plasmids, which will transfer the metabolic products. So basically what is plasmid doing? This exchange between two unrelated bacteria, what is this called as? This is called as horizontal gene transfer. This is what is known as horizontal gene transfer. When the genes pass from mother to child, that is vertical. But you are not related to me, I am passing information to you, unrelated bacteria exchanging information. This is horizontal gene transfer. All right, excellent. So, let's quickly talk about antimicrobial resistance. And you know that antimicrobial resistance is the ability of a bacteria to resist the action to other antimicrobials or antibiotics. And you know that it could either be intrinsic. So intrinsic means that the bacteria has the inherent by birth property to resist certain antibiotics. For example, the antibiotics which act on the lipopolysaccharide layer, all gram-positive bacteria are going to be intrinsically resistant to that antibiotic, because gram-positive does not have a lipopolysaccharide layer, whereas the other is the acquired. What is acquired? In acquired, there is the resistance gets acquired either in the form of small little mutations, or they are transferable as by the transfer of the different plasmids. So what happens in mutational resistance? There is point mutation and the bacteria shows low level of resistance. The level of resistance is low and it is usually to one drug or a single class of a drug. Okay. Suppose example is Mycobacterium tuberculosis. So Mycobacterium tuberculosis develops resistance against either rifampicin or isoniazid. So it is low level resistance to either one drug or one class of drugs. It can only be transferred vertically. Okay, it can only be transferred vertically, only from mother to child, not horizontal between two bacteria. And they can be overcome by drug combinations. So so that is why in Mycobacterium tuberculosis, I give multiple drug therapy, so that even if there is resistance to one drug, the others can take care of the bacteria and keep it at a low level, and I do not risk spreading the tuberculosis like crazy. Yes, so that is mutational resistance. I can just give many drug combinations and that will take care of it. The problem is the transferable drug resistance, because this happens to many classes of drugs at a single time. Okay, many classes of drugs are involved. So at the same time, the bacteria is going to be resistant to aminoglycosides also, penicillins also, then your cephalosporins also. So बहुत problem हो जाती है. That is why it makes the multi-drug resistant organisms. And the worst thing is that they are horizontally transferred. Horizontally transferred. So two sitting bacteria next to each other can become NDROs just by exchanging plasmids with each other. This cannot be overcome by drug combinations, and there is a very high level of resistance which is associated with these transferable drug resistance, and they cannot be overcome by multiple drugs. They cannot be overcome by multiple drugs. So that is why it becomes difficult to treat your multiple MDROs. So the different mechanisms of AMR are enzyme inactivation. So what can a bacteria do? It can release certain enzymes, it can release certain products which inactivate the enzymes. So suppose there is beta-lactamase. What does this beta-lactamase do? This is an enzyme which goes and inactivates the antibiotic. So beta-lactamase will go and break the beta-lactam ring. So it is inactivation of the antibiotic by the enzyme. Inactivation of the antibiotic by the enzyme. Okay. Target site modification. Your gram-positive bacteria, they change the penicillin binding protein into penicillin binding protein 2A. Now your penicillin drugs don't know where to bind on the cell wall, so they cannot break the cell wall. Porin channels, example by the gram-negative bacteria. The porin channels, what do? They don't allow the antibiotics to come inside. They become selectively porous. And then efflux pumps are like vomiting centers. They are like things which expulse out the antibiotic. So either the bacteria will release enzymes which will go and inactivate the antibiotic, or bacteria will not allow the antibiotic to bind to a target only by changing the shape of target, or they will not allow the antibiotic to enter inside the cell. Now suppose everything fails and the antibiotic is able to enter inside the cell also, then the bacteria will activate the efflux pump and they will expulse out the antibiotic of the cell, out of the cell, expulse the antibiotic out of the cell. Right? So it is like a vomiting mechanism. The bacteria will vomit out all the antibiotic outside. This efflux pumps is majorly seen with, example is Pseudomonas. Pseudomonas, right? So we have covered so many important topics today, and what I want you to do as a homework is you go and revise the bacterial genetics. Bacterial gene transfer, the horizontal gene transfer, which is done by transformation. So you have to learn the mechanism of transformation. Very, very important, you learn the Griffith experiment. The Griffith experiment with the mouse, you have to learn it. Then you go and you read transduction, and in transduction you read the two types of transduction. That is the generalized transduction and the restricted or the specialized transduction. And finally, you have to read about the different types of conjugation, including the high frequency recombinant and the HFR conjugation, right? So please go and read these topics as well. So if you have any doubts, you can definitely ping me on my social media, and then I will connect with you soon. And I will see you soon in another important session of FARRE. We are going to cover immunology in a single shot. So stay tuned. Happy learning. Thank you for joining in.